Every second of every day, while you were sleeping and while you were awake, your body's at work creating the proteins you need to survive. These proteins, which are made inside your cells, create muscles, transport oxygen, repair tissue, cool you down, and much more. Your body, a working machine, effortlessly manufactures proteins in a process known as protein synthesis. Protein synthesis is divided up into two parts, transcription and translation, which resemble the copy and paste settings on your computer. Let's begin with transcription. There are two main cell parts that are at work in this step, namely DNA, the genetic material of a cell, and the nucleus, the protective barrier surrounding the DNA. Your DNA is the mastermind behind what proteins your cell makes. In fact, DNA is copied and the information is taken to another part of the cell, called a ribosome, where the protein will be made. You may be thinking, why can't the DNA just go to the ribosome itself? Well, DNA cannot leave the nucleus for the same reason the Secret Service is always trailing the US president. It is so important it can't risk the chance of being destroyed. Now that we've established the fact that DNA cannot leave the nucleus, let's learn about how it is copied. If only there were a copy button in your body, things would be so much easier, you may be thinking. But the cell has its own type of copy button, and it is made up of two things, a promoter and RNA polymerase. Promoter is the section in DNA upstream of the section of DNA that is going to be copied. RNA polymerase is an enzyme that is going to actually copy the DNA. A promoter acts as a flag to tell the RNA polymerase where to begin copying. RNA polymerase scans the DNA until it finds the place where the promoter ends. This spot is known as the transcription start site, or the TSS for short. To understand what is going to happen next, we need to brush up on the structure of DNA. DNA consists of two backbones and four bases, adenine, thymine, guanine, and cytosine. Now these bases pair up with their friends, adenine plus thymine and guanine plus cytosine. What RNA polymerase is doing when it is copying the DNA is it is unraveling and separating the base pairs. Now there are loose bases floating around in the nucleus. Sensing their chance to make friends, they attach to the lonely separated DNA bases. The RNA polymerase then rips off the new strand and closes the DNA back up. As the RNA polymerase moves along, it repeats the process, and the strand grows longer and longer. Soon, the RNA polymerase is signaled to stop. It separates the DNA and the new strand, and then it leaves. This new strand is called RNA, and it is the perfect duplicate of the original DNA. I guess the copy button worked. Well, almost. Remember how we talked about the four bases in DNA? Adenine, thymine, guanine, and cytosine? Well, RNA replaces thymine with another base known as uracil. Also, DNA has two backbones, while RNA only has one. All I can say is disregard the differences because they won't matter in the next step. There are a couple more cell parts involved in the next step, which is translation, namely the cytoplasm, a jelly-like substance in which all the cell's organelles are located, and a ribosome, where the protein gets built. The new RNA, called messenger RNA, leaves the nucleus through a small gap and floats through the cytoplasm until it finds a ribosome. The thing about ribosomes is that they have two halves. The mRNA attaches onto one of the two halves and is sandwiched in by the other, kind of like you sandwich salad in between two tongs. Now it's protein making time. Proteins are made up of a chain of things called amino acids. The order and shape of the amino acid chain decides what protein is being made. The responsibility of putting the amino acids in the right spots is given to another type of RNA called transfer RNA. Now transfer RNA reads messenger RNA in sections of three bases. Each individual piece of transfer RNA carries one specific amino acid along with it. When it finds a piece of messenger RNA that it corresponds with, it attaches to it and connects its amino acid. As more and more pieces of transfer RNA visit the scene, more and more amino acids are dropped off, ultimately forming a protein! Yay! An easy way of thinking of the whole process of protein synthesis is this. There's a document, DNA. The important parts are scanned and copied by a promoter and RNA polymerase. Of course, the promoter just helps. The information, messenger RNA, is sent to a protein-making facility, the ribosome. Next, the factory workers, transfer RNA, follow the directions on the document to bring over the right materials, amino acids. Once they finish all the instructions put forth by the document, they stop, and the product protein is created. All of this is going on inside your body right now. Isn't your body amazing? Want to learn more about genetics? visit DNALC.org. We've only just brushed the surface. Thanks for watching!